Assalamualaikum and hi. Today we are going to do an experiment about SDS page. So, what is SDS page? SDS page is an electrophoresis method that separates proteins based on their mass using a polyacromide gel as the metric. The gel used is a polyacromide-based discontinuous gel, usually prepared as a slab gel sandwich between two glass plates. SDS is added to the sample and it binds to proteins at a ratio of about 1.4 gram of SDS per gram of protein, which equal 1 SDS molecule per 2 amino acid. SDS denatures protein and masks their intrinsic charge, giving them a uniform negative charge to mass ratio. When an electric field is applied, proteins migrate toward the anode, and their speed of movement depends only on their size, not their charge or shape. This process allows accurate separation of protein by molecular weight.
So now, I will show you all the apparatus used in this experiment. First, we have the power supply. The power supply is used to convert alternating current into direct current, which is necessary to run the electrophoresis process. Next, we move on to the tank and lid. The tank and lid are made up of cathode and anode buffer compartments, which are connected using platinum wire electrodes to allow the current to flow. Then, we have the casting stand. This stand serves to hold the gel cassette in place during the gel preparation. Now, we have the clamping frame. The clamping frame is used to secure the casting frame and consists of right, left, and lower parts to ensure everything stays firmly in place. Next is the pipette, which is used to deliver the protein sample into the tiny wells created in the gel. Now, we have three important substances used in this experiment. First, the buffer solution, which is used to denature and linearize the protein. Second, the staining agent, which in this case is combative brilliant blue, used to stain the proteins for visibility. And third, our sample protein, which is the Korean protein. Moving on, we have the plastic comb. This comb is placed into the gel during casting to form the well where the samples will be loaded. Lastly, we have the gel cassette, which is used to load both the gel and the sample protein for electrophoresis.
So now we will move to the procedures of SDS page gel electrophoresis. So this is the sample that we use to run the experiment. And now let's start to assemble the SDS page system and load the samples accordingly. Start by inserting the tall and short gel plates into the casting stand, ensuring that the short plate faces the front. Carefully seal the casting frame to prevent any leakage. Then secure the entire setup into the clamping stand and lock it firmly in place. Next, pour the resolving gel and allow it to solidify. Once it has set, pour the stacking gel on top. Insert the comb into the stacking gel to form wells for the samples. After the stacking gel has fully set, remove the casting stand from the clamping stand and transfer the gel into the electrophoresis tank. The tank consists of two chambers, the inner and outer chambers. Begin by filling the inner chamber with buffer. Gently remove the comb to avoid damaging the gel. Now, load the Korean protein samples into the wells. Finally, fill the outer chamber with buffer to complete the setup.
So now we are going to do a gel staining. First, we have to take out the glass plate, transfer the gel gently into the container, then add 50 ml of distilled water into the gel and microwave it on a high temperature for 1 minute. Then add another 50 ml of distilled water and microwave it for 1 minute. After that, shake the gel gently for 1 minute and add another 50 ml of distilled water and microwave it for 1 minute. After 3 times of microwaving, add 50 ml of Comesi blue staining. Shake gently for 1 minute to ensure even staining, and microwave it for 55 seconds on a high temperature. After that, wash the gel thoroughly with 50 ml of distilled water and shake the gel gently for 10 minutes to remove excess stain. Then add 20 ml of 20% sodium chloride solution. Let the gel soak in the sodium chloride solution for 5 minutes, and after complete, store the gel in refrigerator for preservation.
Switch on the computer and lower part of gel chemidoc. Take out the white conversion screen. Clean the screen using Kim wipe, tissue and distilled water. Put the gel on the screen and insert it into the lower part of gel chemidoc. Click EPI white button for the light source. Click the image lab icon on the computer screen. Choose gel imaging, protein gel, Comesi Fleur Orange. Choose select gel type, BioRed Mini Protein Gel. Set display option, image color, Comesi. Make sure the gel is positioned in the middle of the white screen. Click position gel, run protocol. To edit the gel image, click black or white option. Once complete, switch off the gel chemidoc system. Take out the white conversion screen and discard the gel. Wipe the screen and return it to the storage box.